Thanks for the great discussion. I think we have to move on to our last speaker for today, and I think also the last speaker of the conference. So our last speaker is Nicola Bonzanni. He's working for a company from the Netherlands called Enpicom. Enpicom has taken the task of developing a end-user software for immune repertoire data analysis, and they have recently joined the flock, and they're working with the AIR community to make this software AIR compliant, and have joined several working groups. And we're happy to have them here and talk about what their platform can do, but also about how the development is going and what the ideas are behind it.
Good evening, everyone, and thank you for holding up until now. I'm the last one. So, yeah, I'm Nicola Bonzanni, and what I would like to do in the next 15 minutes is to introduce our company, describe what we've been doing for the past couple of years, and discuss how we think we can contribute to this beautiful community. So let's start a bit with the company. The company is located in the Netherlands, in a beautiful city called 's-Hertogenbosch. And we are currently VC-backed. We are about 20 people, of which 15 are either software engineers or bioinformaticians. So all in all, we are an happy group of geeks that love to develop great code and do good science. And I think this is a key component because software engineers and bioinformaticians have similar skill sets, but also very diverse skill sets. So it's important to combine the two to produce software, scientific software, which is stable and valuable. So what we have been doing for the past year and so has been developing what we call the immunogenomics platform, which is a platform for the management and the analysis of AIRR-seq data. But instead of describing all the different features of the platform right now, I would like to do a step back and discuss what are the principles on which we build such a platform. So let's start from why we wanted to build such a platform. So the kind of request came from the different partners and customers that we interact with. And all of them had one thing in common. They wanted to escape the Excel hell. So they had a bunch of TSV, a bunch of Excel files, a bunch of FASTQ files. And the more study they were doing and the more these studies were growing, the more difficult it was for them to manage those studies and to find the relevant information in these studies. And they had a series of very practical questions that they wanted to solve. Not necessarily so difficult, but difficult for them and cumbersome for them to solve in an efficient way. And some of them are, for instance, identifying and remove contaminants clones or merge samples together, or for instance, identifying public clones across multiple data sets. So those are all relatively simple things to do, but very difficult things to do if you don't have a managed way to handle your data. And all these questions kind of fall into the umbrella of data handling and the management and the integration of these different data types. So we sat down and we thought, okay, how we can solve this problem, or how we can try to solve this problem. And so what we end up with is a simple idea, which is in a way is not revolutionary, linking sequencing data with metadata and external data, but it's very often hard to get it right. So I think we really thought through how can we actually make this link efficient and simple for user to actually adopt and use. And not only we wanted to link to the sequence, the metadata, but also external data. We wanted to link, for instance, other experimental results like ELISpots or whatever other things, for instance, affinity that you are measuring in the lab. And to do so, we started thinking, what are the principles that we can use to make this linking? And we wanted to have certain principles which are simple enough to be easily understood, but flexible enough to encompass all the different or most of the different use cases that people approaches while analyzing AIR-seq data. And to do so, we thought, okay, the first principle is how we're going to group clones together. On the one end of a spectrum, we have a rigid sample definition in which we just have a series of samples, sample 1, sample 2, and each sample contains a number of clones. On the complete opposite side of the spectrum, we have a pile of clones on which we basically remove the idea of samples that we could search through. What we wanted to have is something in between, something which is flexible enough to remove clones from it or add clone to it, and then move this collection of clone in downstream analysis. So the idea, the very simple idea of a clone collection. And what a clone collection allows you to do is a series of relatively simple but also complex operations. So for instance, those are some examples. So if you start from a sample or yellow sample on top, that's the original collection that you find from your biological sample, the initial collection of clones, you can think that you want to subset that sample, for instance, remove a singleton or any other QC filtering that you want to perform. And then you want to create a different collection, which is just what you want to retain from that sample. And you want to move that collection for downstream analysis. And why we want to do that, why we want to create another collection is because we want to keep provenance of the different filtering that you have done, because you want to reproduce this analysis. So the idea is the provenance of the collections is quite important. At the same time, you can perform very complex operational collections. So you can start saying, okay, I have sample yellow, sample blue. I'm going to subset the two samples for QC, that I'm going to join the two samples together in a larger collection, that I'm going to intersect that collection with another one, and just taking the intersection and move the intersection along in the analysis. So this is the first concept. So the concept of a clone collection. And then on top of the clone collection, we said, okay, how are we going to now link all the data and the metadata on these two elements, the clones and the clone collection. So this is not a cell, but it's a clone collection, which contains all these blue clones. And what we're going to do, we're going to add tags to both clones and collections. And what are tags? Tags are key value pairs, and those are structured, which means that a single tag could be, for instance, age, or could be affinity, or any other elements that you think is relevant for your research, could be also gene expression for the matter. And you can start using all these different tagging that you have done and associated to the collection and also to the clone for filtering and searching. So you can perform queries that in one go from, for instance, take elements and values and search through tags that are at the collection level, but also the single clone, so finding specific clones in each collection. So here, I don't know if it's possible to read it from far away, but I want to give you a glimpse of what actually such a search could look like. So the idea is that we can perform very fine-grained searches, and also we wanted to spend some time in actually identifying a good way, an easy way to perform such searches and create queries graphically. So in this case, each line is a single predicate of your search, and we can have searches that are both at the collection level, like you can see here, but also searches that are at the clone level. So we can, for instance, find specific J gene and specific B gene. We can combine the different predicates with logical operators like AND and OR. And we can also do like overlapping searches, so finding through all different collections, all the clones that share the same CDR3, for instance, like in this case. So that allows to perform very broad searches across multiple studies, across multiple collections, but also defining specific clones that we want to find the searches, including also, for instance, clone frequency. An important element here is that if your tags are structured, so they are not just piece of text, but they have a type associated to it, you can do also searches that are specific for the type, like for instance, greater than, lesser than, and so on. So it's very specific also to the kind of data that you have in your tags. Now, so far I told a bit about the idea of having a core of the platform, which is built around managing the data and be able to retrieve the data that you are interested in. But at the same time, we wanted to have the platform to be modular. So the idea is that we have different applications that can use this core. And so far, we developed two applications, one which is AJAX profile, which is our version of annotation from FASTQ file to clone counts, which is able then to read the FASTQs and put those into the platform. But we have also a series of analysis. Some have been shown in the past days, which are in the other application, AJAX Explorer. Now, I don't want to go in the details of these two applications, but I want to show you a key component or a key feature of the platform. So we want, again, to make the platform really easy to use for researchers, also for people that don't have perhaps specific bioinformatics expertise. So the idea is that we want to provide a way, for instance, to select clones directly from the plots. Like what happens in this case, this is one of the visualization of IGX Explore, which is a clone overlap plot. So the counts of each clone, each dot here, as for the different samples. One sample is on the y-axis, another sample is on the x-axis. And then what you can do is directly on the plot, select the clones that you are interested in. And now you can use the concept of clone collection to create a derived collection from those clones. And you can, for instance, prioritize this clone for downstream analysis directly from the visualization. Now, another element that I would like to stress, as I said in the beginning, is we really want to build stable and efficient software. For that, our software engineer have a full development cycle, a stable development cycle, which includes unit testing coverage, as well as continuous integration and several elements that allow us to have a release cycle, which is predictable on the one end, but also is flexible enough to have intermediate updates of the platform. And there are a few, for instance, features that we are already in the plan for the next release that is planned in September. And one key element, again, is the strong versioning of all apps, again, to enable reproducibility. So you can choose whatever version you wanted before of each specific analysis. Now, as I said, the application is modular. That means that you can communicate with the core using a well-defined set of APIs. And those APIs allows, of course, for us to build some of the applications, but also we want to open this API and make them available for the community to build whatever application they want to build that is able, for instance, to include new clones or new collections into the platform, as well as doing and performing specific analysis using the capacity of the platform to identify and extract the correct clones and the correct collections. So finally, I would like to discuss why we think that we should be involved in this community and what could be our contribution in the AIRR community. And I think, first of all, we are already involved in a couple of different working groups that we want to continue that collaboration, and actually we want to increase the amount of contribution that we are having in these two different working groups, as well as we want to incorporate in our platform the MyAIR standards, and we want to also to expose the API for exporting and importing as the AIRR community is defining. And finally, we want to contribute to this different working group in two ways. One is via knowledge. So what we want to do is to share our expertise that we build up, for instance, creating data sets for the benchmarking, as well as contributing in code. We are planning to release a series of tools that we develop internally to build synthetic data sets. And this is something that is going to happen in the next few months. So finally, if you have any questions later on, or you want to see how the platform works, and you have any other specific question, please contact me. We will be here all night. And finally, we are hiring. So if you want to be involved directly, just contact me. And I'm happy to take any question.
All right, thank you. That was a great talk as well. We have time for about 3 to 4 questions. So if there's anything you want to talk about right now, and afterwards we're going to have the reception with the industry and the sponsor, so we can also continue our discussions there. Over there, Scott.
Hi, thank you. So I'm Scott Chrisley. I'm in some of the working groups. I really like that slide where you showed the sort of complicated query with the fields and ors. That's the best visualization I've seen so far of complicated queries. So so the API, the AIRR API, which is being developed, allows complicated queries like that. So I'm really interested if part of what you say you were hoping to share is some of that that visualization, because it looks like a user could do that. I didn't see visually how you constructed it. Maybe you could talk a little bit about that, and what do you think?
Yeah, I think that's, as I said, we put a lot of effort in trying to find a seamless way for user to query the platform. And that's, unfortunately, there is no video that shows how we build that specific query, but it's very simple in terms of you just have a prompt of tags that you have in your data. You can enter your tag, and then you enter the value, and then you choose what you want to match for, and then you can drag and drop those different, let's say, predicates to build these groups. So it's very, I think, intuitive to make, but of course, it would be nice if people try it and can tell whether that's the case. And regarding the API, of course, I think this would be definitely a perfect, let's say, implementation, because you have the interface that allow you to actually build a query, and then if you have your API that expose this, how to grab the data actually that correspond to that query, then you combine the two things. So that's exactly what we are aiming for.
What does the technology stack look like?
Yeah, so the backend is Kubernetes. So basically, each element runs in its own Docker container, and even the applications have their own completely isolated Docker container, which talks in a secure way with the rest of the application. And that allows you not only to have whatever language you want, or whatever, let's say, stack you want in each single application, but also the visualization can be, basically, there are no constraints there. You can have your own specific application built inside that specific container that you're building up. In terms of what the platforms looks like from the front end, and also from the back end of the, let's say, the graphical part, web server, that's Node.js with TypeScript. The database at the moment is a combination of a document database for the tagging part and the relational database.
So related to that, are you planning and digging into what we have for the AIRR-seq database, for example, that if people are releasing their repositories, that you would be able to query it using your IGX platform?
So you mean the part that AIRRC4 query our database, or that we query AIRRC4?
Well, if it's your database or the customer's client's database, that might be difficult, obviously. But if they want to release their data,
yeah.
that would be.
Yeah, so basically that's one of the things that we are going to implement in version 1.1 is the ability to define workspaces. And workspaces then will be also the option to be exposed externally. And workspaces are going to use tags again. So you're not forced to expose a complete study or not even a complete sample. You might choose to expose even single clones, tagging them with a specific workspace.
I was just curious, how do you define a clone in the software?
Yeah, that's a good question. So right now, it's mostly, actually the correct definition would be a chain, because also we are not at this moment having the pairing information that will be released in the next one, a possibility to link both chains and then have a complete clone. And as we define it right now, it's just by CDR3 identity, but that's one thing that we want to make customizable, so that you can actually come up with your own definition of clone.
Thank you for the presentation. So you were talking about external data. Mm-hmm. So can you tell a bit more about this? So what kind of data are you adding right now?
Yeah, so there are, I think, at least two interesting use cases. One is when for data, we mean, for instance, affinity. And then we can import that data and tag that the different clones with the different affinity. So that's data generated in the lab. But another case of external data is external database in terms of clone database, which have been, for instance, annotated for specificity. And then if you can import, and you can import that data inside the platform, then you can start making interesting queries in which you are looking which kind of the clones that you found in your own studies matches the one that has been already annotated, for instance, in an external database.
All right, if there are no further questions, I think we have some announcement to make before going over to the reception. We do, all right.
Why don't